of experience in the field, Dr. Fuat is the ideal person to deliver today's talk. Dr. Fuat will be presenting on Elementary in Fungal Identification Part 1. Before we start, just a quick note on housekeeping. If you have any technical issues during the session, please use the chat function to reach out to our support team. During the presentation, all participants will be muted to avoid any disruptions. You can post your questions in the Q&A box and we will address them during the Q&A session after the presentation. The link for attendance will be shared later. You may check at the chat box or can scan the QR code, which will be shared later. Without further ado, I'll hand over the virtual floor to Dr. Fuat. Dr. Fuat, Doctor may present whenever you are ready.
Okay. Assalamualaikum warahmatullahi wabarakatuh and very good afternoon. Thank you, Dr. Jayamalar, for the kind introduction. Thank you, the organizing committee of MSIDC, MMIG, for the top invitation. Thank you to all participants for your interest in the topic. As we all know, fungal spores germinate. I'm sorry. So this is topic of today's presentation. As we all know, fungal spores germinate and form mycelium. This mycelium grow on the surface of the agar and then penetrate into the growth agar to form substrate mycelium. There are vegetative hyphae. More and more substrate myceliums are formed and more of the myceliums on the surface grow and form aerial mycelium. This aerial mycelium gives the fuzzy appearance of a Mohd colony. Over time, such as 1 week, some fertile aerial hyphae may form a special reproductive hyphae, such as conidiophore, sporangiophore, or pylites, and conidia or spores are formed. Mating of some fertile hyphae may also form sexual fruit bodies, and the fungus are readily to be identified. If we prepare slide of the colony, when the reproductive hyphae have been produced, we will see conidia, spores, or fruiting body. We will jump happily because we know we may be able to identify the fungus. However, this is not always the case. The first time we prepare a slide, the colony is probably in this state where there is no spore, although the culture has been more than 1 week old. Or we may observe arthroconidia or chlamydoconidia, which are common to a lot of fungi. We then incubate this culture for another week, hoping the culture will sporulate in the next 1 week, but it did not. Because of the TAT set by the ISO document, the growth of agar has shrunk, plus the pressure from the clinician, we may report the ID as non-sporulating mode, or we send the culture to a reference lab. We are happy, but not the clinician. Before we conclude that this fungus does not sporulate and no ID could be given. You should observe the colony in more details, as it may show some clues to its identity. Lab should also keep this information in their worksheet and produce it to the reference lab. You may be surprised that you could actually identify the fungus to at least into its group, such as Basidiomycete, Zygomycete, Omycete, etc. So this is the focus of this presentation today, and I will cover the following fungi: Mucormycete, Zygophagomycete, Oomycete, especially Pythium insidiosum, Glomeromycete, Basidiomycete such as Schizophyllum communis, non-sporulating dermatophytes, Coccidioides, Malbrancher and Malbrancher-like fungi, Neoscytilidium, Scytilidium, Onychocoli and Atrographes, Atroconidal forming filamentous yeast, black yeast Aureobasidium pullulans, and lastly, dermatophytes with no macro or micro conidia, but with unique characteristic of Clamidoconidia. So if you prepare a slide, regardless of what age of the colony, as long as you can see the colony, you will definitely see a hyphae. So the first important information is from the observation of this hyphae, and it is whether the hyphae is hyaline or the multiaceous. So hyaline hyphae means it has no it is, I mean, if you stain the hyphae, it will be blue, it will stain to blue. If you stain the hyphae, it will be red, it will be red. But the multi-acial hyphae has pigment in its cell wall. So this pigment is actually brown. So it make the hyphae looks brownish. So brownish hyphae is called the multi-acious hyphae, and the fungus which show this the multi-acious hyphae is called the multi-acious Mohd. So shown here is the hyaline hyphae on the left, stained with LPCB, and on the right, the fungus was stained with LPCB2, but it didn't pick up the stain and remain as its original color, that is brownish. However, sometimes it's difficult to determine whether the fungus is the multi-acious or not based on the color of the hyphae, but you can confirm the fungus is the multi-acious by looking at the colony, its colony, because the multi-acious fungus colony will be brownish too, either from the top or from the bottoms of the agar plate. Actually, there are a lot of the multi-acious fungus isolated from human diseases. So shown here are some of them. So I may in future discuss fungus in this group, but please, I would like to stress here, please, if you do identification of mold, you must write, first of all, in your worksheet, whether the fungus you see, you're studying is hyaline or the multiaceous. Even though if you do not identify the fungus, you're sending the fungus to a reference lab, but you must keep this information in your worksheet, because when you receive the ID from reference lab, maybe they are using DNA analysis, DNA sequencing, so MALDI-TOF, they will send you back with the ID, and you can confirm whether they have analyzed your original culture by confirming. Let's say if they report to you Acrimonium species A, then you can go to Atlas, Mycological Atlas. Atlas of fungi will always tell you whether the fungus is hyaline or tomateaceous. So this is one way how you could verify the results you receive from the reference labs. I know that Malditov is getting better and better now. DNA sequencing is almost perfect for identifications of fungus, but you may send culture to them, but something else grew and they identify different culture from what you have sent to them. So it's very important, first of all, when you identify fungus, whether you see the spore or not, make note in your worksheet whether there are hylines or tomateaceous. This is example, Acrimonium in Mycology Atlas. So it says here, hypae are fine and hyaline. For pure Acrimonium parasiticum, hypae hyaline, later becoming tomateaceous. So actually, pure Acrimonium is tomateaceous mode. Secondly, when you study the fungus under microscope, whether you see spores or you don't see spores, you must also take note whether the fungus, the hypae, has septum or hardly, you could hardly see any septum there. So the hypae with, you can see a lot of septum, is called septate hypae. And when you don't see septation or the septa are far apart, sparse, so we call this one synocytic hypae. If you look at this flow chart, this is quite old classification of kingdom of fungi based on sexual or asexual reproductions. So you see here, there are two groups of fungus which have mycelium aseptate or synocytic. There are zygomycete and the other one is oomycete. So zygomycete, I think for the last 6 or 5 years ago, this zygomycete is quite simple group. When you say zygomycete, they are either mucorales or entomopterales. So mucorales group cause mucormycosis and entomopterales cause entomopteromycosis. So if you see it's synocytic hypae, then you can easily report the declination that at the moment you identify the fungus as zygomycetes. But now, this zygomycetes has been divided into two groups. So we don't call this fungus in these two groups zygomycetes anymore. They are either Mucoromycota or Zoophagomycota. So Mucorales fall under Mucoromycota and Eptomothoralis fall under Zoophagomycota. So anyway, whether there are more groups, there are more genera now in this Mucoromycota and Zoophagomycota, but one feature that's shared by all these groups are their hypae are synocytic. So this is the most important group with synocytic hypae group under Mucorales. And probably you have heard about Syncypalastrum, Saxenia, Cunninghamilia, Mucor, Rhizopus. So all these important zygomycete are in the Mycorrhizae group. Okay. So I have a question here. What are the chances of a fungal isolate with broad hyphae to be later identified as species of Mycorrhizate or zygomycete, both formally called zygomycete? Okay, zygomycete not only show synocytic hyphae, but broad hyphae. So if you see synocytic hyphae only, you can't jump to a conclusion that you are looking at zygomycete or now mucormycete or zoopagomycete. Secondly, after you see that the fungus show synocytic hyphae, the next thing is to see whether there are broad hyphae or narrow hyphae. I think so. Zygomycete has broad hyphae, so their hyphae is at least 7 micron, up to 30 microns. So they are synocytic and also broad. So if we see synocytic hyphae and broad hyphae, what are the chances of this fungus to be identified as Mycorrhizate or zoopagomycete? Okay, that's the question. We will answer this question together after I show you a few more slides. So apart from zygomycete, there is another big groups of fungus in the group. We call it oomycete. Okay, oomycete. So oomycete too has synocytic hyphae and fairly broad hyphae too. So I think the most important oomycete is Pitchaimuthu insidiosum. So Thailand has many cases of this. I'm not sure whether we in Malaysia has isolated Pitchaimuthu insidiosum, but this is very important oomycete. So if you look at the growth on the blood agar, so the colony is soft, glabrous, submerged or radiate white yellowish colonies, and are usually detected in less than 24 hours. So this is very important difference between Pitchaimuthu and zygomycete. So their colony is glabrous, but in zygomycete, they are woolly. Okay, and the rate of growth also, this is too fast for zygomycete. So microscopically, hyphae are sparsely septate, that means synocytic, and this is the waist of the hyphae. So it is smaller than zygomycete, with irregular but mostly acute angle branches. So acute angle branches. Okay. So their hyphae is slightly smaller, even though they are still considered broad, but smaller than the hyphae of zygomycete. But compared to aspergillus, they are broader. Aspergillus, as well as candida, so the waist of the cell is 2 to 3 micron, but Pitchaimuthu insidiosum 3 to 6 micron. Okay, that is Pitchaimuthu insidiosum. I hope maybe one day we can isolate this organism. I wouldn't say it is a fungus, this organism from our clinical specimen. So more on this fungus. So the proper identification of culture is done by the formation of flagellated zoospores using the Mendoza and Parandas techniques. Using water culture or by DNA analysis. So if you want to further identify this organism, you should do water culture, because in water culture, they will form zoosporangium. So we don't see this in zygomycete. And this zoosporangium will release zoospores. So this is again, it's not in zygomycete. So for comparison, Pythium isodiosum versus Mucoromycete or Zoophagomycetes. So their colony morphology and rate of growth are different. And in terms of ways of hype, Zygomycete broader. And another important aspect is that if you want to culture Pythium, your sample collection must be in water and transported to the lab within hours, less than 1 day. So I don't think the way that we send specimen to our lab today, we will culture out Pythium insidiosus. Just now I said I don't call oomycete as a fungus because oomycete is actually not a fungus. So this is all classification. Oomycete is not a fungus, but strangely they behave, they look like very similar to fungus, very similar to mold. Okay, they can be cultured on agar plate, but to identify them, you need to use water culture, and the specimen collection and transportation must be correct. So apart from oomycete just now, oomycete, is there any other group of fungus that show cynocytic hyphae? There are actually. It's glomeromycota or glomeromycete, and also chytrids, chytridiomycota. So we're going to look into these two groups. So this is the 5 phylums of fungus. Okay, these two contain most of the fungus that we isolate in the clinical specimen. Just now we talked about zygomycete. So now we're going to talk about glomeromycota. So like most fungi, the glomeromycota generally exists as filamentous hyphae. And like the zygomycete, such as bread molds, the hyphae have no closed wall. Their members are cynocytic. So glomeromycetes are fungus, and they are cynocytic fungus. So, but glomeromycota are obligate symbiont, meaning they cannot survive without their photosynthetic partner. Consequently, they cannot be grown in lab unless they are provided with a suitable host. That means if you want to grow glomeromycota in the lab, you should put inside your media some plant tissue so that they can grow into the plant tissue. Thus, they are very unlikely to be cultured on routine mycological media. So shown here is plant tissue or fragment, and inside there we see cynocytic hyphae. That is the hyphae of glomeromycete. So they grow symbiotically with the plant. This is another slide showing synesthetic hyphae in plant fragment. So the next is Chytridiomycota. Chytridiomycota, they are fungus and they show synesthetic hyphae if present. So what makes they different from other group of fungus is they have multi-zoospore in the asexual mode of reproduction, and in sexual modes of reproduction, they produce sporangiospores. So this is the basic general morphology of chytrids. So they have thallus, the main body of the chytrid, in which most of the cytoplasm reside, and from which system of branching elements emerges. So the latter is called rhizoids. Rhizoid. So the rhizoid anchor the fungus into its substrate and secrete digestive enzymes. So the thallus is converted into sporangium during reproduction. So the sporangium is sac-like, and its protoplasm becomes internally divided to produce multi-zoospore inside here. So most chytrids usually live in aquatic environment, so not in land. Most chytrid usually live in aquatic environment, and chytrid reproduce both sexually and asexually, which leads to production of multi-zoospore, which we don't see in zygomycete, for example. And chytrids are mostly unicellular. So we come back to this question: what are the chances of fungal isolate with broad hyphae to be later identified as species of Mycorrhizamycete or Zoophagomycete? So the answer is, my answer actually is highly probable. Okay, now we go to another groups of fungus, Basidiomycota. So Basidiomycota here, they have septate hyphae. They have septate hyphae, and we normally call this group of fungus as mushrooms. So these include toadstool, shell fungi, bracket fungi, and coral fungi. So mushroom or Basidiomycete, if you culture them on our routine mycological media, they don't form fruit body. I mean, they don't form spore. So most of them, majority of them won't form any reproductive structure when grown on routine mycological media. So what you may see in Basidiomycetes is clam connection. It is a hook-like structure formed by growing hypocell and act as hyperbridge between two cells. So these are the clam connection. So under laboratory condition, Basidiomycetes from clinical specimen usually present as non-sporulating molds that grow as cottony white colonies. Colonies that may emit an unpleasant odor, such as bleach-like odor in S. communi, are susceptible to cyclohexamide and are tolerant of the fungicide vinomyl, and hyphae with or without connection are possibly Basidiomycetes. That means if you see septate hyphae fungus, but no spore, you may see clam connection or you may not see clam connection. But if they show cottony white colonies and some emit an unpleasant odor, and they are susceptible to cyclohexamide and this fungicide vinomyl, so that fungus maybe basidiomycete. But if you see clamp connection, that is confirmed that the fungus you're looking at is basidiomycete. So if you don't know how to identify basidiomycete, because basidiomycete will usually identified by studying their sexual spores or fruit bodies, but actually you can report to the clinician that the fungus is basidiomycete. So until the reference lab where you send the culture give you the definite species of the basidiomycete. So this paper published in Isham's journal, recognizing filamentous basidiomycete as agent of human disease. So basidiomycete is mushroom, but there were reports of this fungus isolated from human sample, the clinical samples, and proven to be the agents of the disease. So in this report, 50% of the basidiomycetes in clinical specimen were Schizophilum communis, and about 6% Homographiella aspergillata and 5% Seriporia lacerata. So we have other genera of basidiomycete as well, but 60% of the basidiomycetes are in this group, Schizophilum communis. So clamp connections are considered characteristic of basidiomycete and may be observed in as many as nearly 80% of clinical isolate, especially in Schizophilum communis and Seriporia lacerata. So for non-sporulating and white woolly colony, please observe for clamp connection, the fungus could be basidiomycete. So more on Schizophilum communis. So Schizophilum communis not only show clamp connection, but please bear in mind that not all Schizophilum communis will show clamp connection, but for this culture, it shows clamp connection. And there is another important features of hyphae of Schizophilum communis. It also shows this structure. We call it spicules. Spicules. This tiny thorn-like structure, spicules. So clamp connection plus spicules, then it's probably Schizophilum communis. So this is Seriporia lacerata, also shows clamp connections. Okay, now we come back to this colony of fungus on mycological media, but this colony do not produce spore or not yet producing spore. So what we can see if we prepare slide from this colony is first of all, we definitely will see this hyphae, mycelium, either hyaline or dermatiaceous, and we may probably see this vegetative hyphae. They are not reproductive hyphae, but they in the special shapes. So this is what we call it spiral hyphae. This one, like the tooth, is called pectinate body or pectinate hyphae. This is fabric chandelier or antler hyphae, nodular organ, and ragate hyphae. So these are not reproductive structure, and these can be seen in a lot of fungus. They are not specific to any species. But they probably useful in the identifications of some dermatophytes. As we know, dermatophytes are pathogenic fungus, but not all of the metophytes produce macro and microconidia. So when we learn about identifications of the metophyte, rubrum, metatherophyte, for example, tonsurans are differentiated based on their microconidia and macroconidia. But there are several dermatophytes, like here, okay, here, and here. So they are important dermatophytes, even though they are isolated less frequently compared to Trichophyton rubrum, Trichophyton metagraphite, Trichophyton tonsurans. But I had actually during my working days in IMR, isolated this dermatophyte. So no microconidia, no macroconidia, but we could identify this fungus by studying their hyphae. So this is Trichomata metagraphite with spiral hyphae. So you see spiral hyphae, but this spiral hyphae is not necessary to be seen for you to identify this fungus as Trichophyton metagraphite. So don't wait until you see spiral hyphae, then you make decision that you are looking at Trichophyton metagraphite, because Trichophyton metagrophytes are identified based on the microconidia. So, but look at this Trichophyton Schollenii. So this is Trichophyton that usually don't produce macro or microconidia. But under microscope, you see this what we call it fabric chandelier vegetative hyphae. And if we see this one from a culture isolated from, let's say, a nail or skin, then look at the colony. If the colony is light gray, waxy surface, irregular border consisting mostly of submerged hyphae, the surface of the colony is usually non-pigmented or tan, furrowed and irregularly folded, then this culture is probably Trichophyton Schollenii. Another Trichophyton that we could identify based on the vegetative hyphae is Microsporum auduini. So this Microsporum, microconidia are rarely produced, but they show comb hyphae. This is comb hyphae. So if this isolate is from skin or nail, and their colonies are flat spreading, grayish white to light tan white in color, have dense white light to downy surface, and rivers can be yellow brown to reddish brown, so that the culture is probably Microsporum auduini. Trichophyton southernensis also can be identified. First of all, look at the colony. So it has deep apricot orange color. And under microscope, you don't see all that fabric chandelier, for example, but you see hyphae often show reflexive and right angle branching. Either right angle or reflexive means the direction of the hyphae is opposite. Normally the branching is on the same direction, but in southernensis, they have reflexive branching opposite to the adjacent branch. So this is quite specific actually for Trichophyton southernensis. So I think we had one case in RCPA exercise that sent Trichophyton southernensis. So it's quite easy to identify it if you study the hyphae and you see the reflexive hyphae, then look at the clinical summary. If it is from the dermatological specimen, then it's Trichophyton southernensis. You don't have to wait to see microconidia or macroconidia when you identify Trichophyton southernensis. Microsporum ferruginum too. So these also dermatophytes in the groups of Microsporum. They don't also produce macroconidia, but if you see irregular branching hyphae with prominent crosswall, we call it this bamboo hyphae. So this is a characteristic of Microsporum ferruginum, providing the colony characteristic similar to what is described for Microsporum ferruginum. Okay, now we move to, because I think we are running out of time. So we move to vegetative reproduction. So even though there are vegetative hyphae, but certain hyphae, especially in the aerial hyphae, they are fertile. They can reproduce. So we call this one vegetative reproductions. So in this presentation, I would like to focus on two methods of reproduction by vegetative hyphae. There is fragmentation and formation of chlamydospores. So this is how arthroconia is formed. You know, this is the hyphae. It elongates and then it forms septation. So there are so many cells in the hyphae. Each cell transform into spore, or we call here arthroconidia. So this is again is common in a lot of fungus, and it's probably the only conidia that you can see when you try to identify many modes. So actually you can identify the mode if you see only arthroconidia. So but there are some fungus that you can identify them based on their arthroconidia. So these arthroconidia, they are either single cell, one cell, or two cell arthroconidia. And there are two types of arthroconidia. One with disjuncted cell. The other one is without disjuncted cell. So without disjuncted cell, all the cell will turn into arthroconidia. But if the fungus form arthroconidia but with disjuncted cell, then the arthroconidia normally is inalternate because the cell in between them does not turn into arthroconidia, and lysis of the cell will disperse the arthroconidia. So if you study a fungus and you see arthroconidia, so first of all, check whether it has a disjuncted cell or without disjuncted cell. Because if it has disjuncted cell, so it could be coccidioides immitis or coccidioides posadisci, but we don't have this fungus in Malaysia. I did isolate this fungus, but the patient was from the US. But it's important to understand or to study this hyphae arthroconidia with disjuncted cell, because we may culture Malbranchia. Malbranchia is the closest fungus. Looks like very similar to coccidioides. It has disjuncted cell. It produces arthroconidia with disjuncted cell. In coccidioides, the arthroconidia has been described as barrel shape, whereas in Malbranchia, They are rectangular with truncate ends. Truncate means broad ends, okay, broad ends like this. So this is truncate end, Acroconidia. So how do we differentiate the coccidioides from branchia? So coccidioides Acroconidia usually shorter but wider compared to branchia. So there are several other genera of fungus which look like Malbranchia. So we call it Malbranchia-like fungi. So this report was published in IMA Fungus Journal. So apart from Malbranchia, we have Pseudomalbranchia, Arachnomyces, Spiromastigoides, Curamyces, Pseudoaptrosis. These are names of fungus which look like Malbranchia. So that means they look like coccidioides too. And we thought that Malbranchia are saprophytic. So 20 years ago when I isolated Malbranchia from clinical specimen, I won't report it to the clinician. But maybe nowadays we have to look at the clinical information. So whether we should report Malbranchia or Malbranchia-like fungi, because they have been implicated in mycosis elsewhere. Okay. So I want to discuss about this Malbranchia-like fungus, but you can see here they produce Arthroconida with disjuncted cells. So Arthroconida, without disjuncted cell, we have several important fungi from this Arthroconida, but without disjuncted cell. So first of all is Scytilidium. Scytilidium, the Mycology Mohd. So they produce Arthroconida. And another important dermatiaceous mode that produces Arthroconida is Neoscytilidium denidatum. It was Scytilidium before, but now called Neoscytilidiums. So this is the most common Scytilidium fungus. You could isolate from diseased nails. It's almost 100%. Probably 90%, 95% of fungus is a snail, and you isolate a fungus. It is demarcaceous, and the fungus most probably is Cryptridium demidatum. It's not difficult to identify this fungus because it produces Arthroconida, and you see some of the Arthroconida are two-cell with dark septum. So this is the characteristic of Neoscytilidium denidatum. And this is how the colony looks like. So they are the multi-ashes mode. Okay, these photos are taken from a publication. So this author show skin scraping, and in the skin scraping you can see these two cell. This the multi-ashes Arthroconida, and you can see two cell Arthroconida. So when the author cultured the specimen, they grew this, and you can see this two cell Arthroconida. So it's confirmed not only by culture but also by direct microscopy. I think I would like to stress, if I ask participants, I think many of you you don't do this direct microscopy observation when you studying a case, mycosis. You culture straight away the specimen and try to identify the organism. You could do that, but it's sometimes quite difficult to determine the significance of the culture that you isolate. But if you do direct microscopy, then from here you see the multi-axis mode. And secondly, you see two-cell arthroconidea. And when you culture the organism like this one definitely came from this. So that means this fungus is significant, even though you don't know what fungus is this, but you see the spore in the specimen, you see the same spore in the culture. So this is already a significant fungus. If you see cell which look like Scythidium dimidatum, new Scythidium dimidatum, but they are highline. They are not multi-axis, but look similar microscopically to the Neoscatridium dimidatum. So this is another species of Neoscatridium. We call it Neoscatridium here, linums. So I think if you isolate Scatridium from specimen, 90% of the time you probably will isolate Neoscatridium dimidatum. That 5% is probably from this species. This is fungus which can be isolated from nail infection. So it produces arthroconidea, chain of arthroconidea. But if you look in more detail, look carefully. So they are not the multi-axis, they are highline, but they also have two-cell arthroconidea, and usually you can see constriction at the septums of that two-cell arthroconidea. That's how we identify Onychocola. And if we have the colony, we can confirm it as Onychocola canadensis. This is another fungus. When you prepare slides of this fungus under microscope, you probably can see chain of arthroconidea again. But this arthroconidea is not, mostly it's not from the hyphae, but the hyphae produce a special reproductive hyphae. We call it conidia 4, and the conidia 4, the arthroconidea form from the conidia 4. That's why you see this part here not turn into arthroconidea. So this is Arthrographis, and the important species of Arthrographis is Arthrographis calcare. So Arthrographis calcare, on the fourth day, if you look at the colony, they look similar to Candida albicans. And if you do teeth mount or wet mount, you see a lot of yeast cells. So you may think this culture is Candida albicans, but if you incubate the culture further, on the 10th day, it turns into Mohd. And under microscope, you can see a lot of arthroconidea at the tips of conidia 4. So there were many cases of fungal infection caused by Arthrographis calcare reported. Okay, now we move to Trichosporon. Trichosporon are filamentous yeast, and they produce arthroconidea. I think Trichosporon is very important yeast. in terms of antifungal susceptibility, because they resistant to many antifungals. So it's very important we can identify these fungus correctly. So they look like yeast. If you look at the colony, they initially they look very similar to yeast colony. We call it yeast-like colony, but later they become drier. Initially, most of Trichosporon will show yeast cells, but later on the form hyphae and the hyphae disarticulate into arthroconidia. Okay, one of the important Trichosporon is Trichosporon asahi. So colonies are white to cream colored, powdery. So this is not yeast characteristic. Okay, Swat-like, so more like a mold with radial furrows and irregular folds. So in culture, they appear as yeast cell, which grow to develop barrel-shaped arthroconidia. I think there's something strong with this sentence. Actually, in culture, initial culture is correct. Initial culture appears as yeast cells, but then later on, they produce a lot of hyphae, true hyphae, and that hyphae turn into arthroconidia like this. This is the true hyphae, and this is the arthroconidia form in Trichosporum asahi. So this Trichosporum was isolated from blood culture. So blood culture, in blood culture, you can see blastospores, and you can see also pseudohyphae and also true hyphae. Another important species of Trichosporum is Trichosporum ovoides. So in this isolate, budding cells are absent, arthroconidia are cylindrical. Again, Trichosporum incane. So here we see true hyphae and lots of arthroconidia, and we have this structure. We call it aprosoria. Actually, this is hyphae. But anyway, this is not specific to Trichosporum incane. Okay. Initially, 4 or 5 years ago, maybe 4 or 3, at least 3, before 3 years ago, if you see yeast-like organism, and under microscope you see true hyphae, and you also see arthroconidia, then you can think about these 3 organisms. Okay? You see true hyphae, there are 3 possibilities, either Trichosporum, Geotrichum, or Yerrovia. But if you see through hyphae, yeast-like organism, colony yeast-like, but no arthroconidia, then you can rule out Yerrovia. So it's not Yerrovia, it's either Trichosporum or Geotrichum. And we can differentiate Trichosporum from Geotrichum by doing urease tests. Because Trichosporum, urease positive, Geotrichum, urease negative. But recently, there are 2 more genera of yeast, hyper yeast, with arthroconidia. Okay, but they are not Trichosporum. Okay, but they have true hyphae, they form arthroconidia. You may see budding yeast, and urease is positive, but they are not Trichosporum. So they are either cotuneal Trichosporum or Epiotrichum. So So now, for correct identifications of filamentous mold, you need to do a complete identification like API, MALDI-TOF, DNA sequencing, because by morphology, now you can definitely say that when you see through hype, atroconidia, urus positive fungus, then they are not necessarily trichosporon anymore. Okay, lastly, we will be talking about chlamydospores. So the formation of chlamydospores are similar to the formation of atroconidia, but the spore, the cell wall of the spores or chlamydospores is thicker, and they usually darker than the hyphae. So, and in atroconidia, these atroconidia are readily disarticulated, but not in chlamydoconidia. They remain intact like this. So that is the differences between atroconidia and chlamydoconidia. So there are several terms regarding the chlamydoconidia. If they are positioned at the end of hyphae, we call it terminal. If in the hyphae, we call it intercalary. Side of hyphae, we call it sessile. Okay, sessile. So chlamydoconidia walls are thicker and usually darker than walls of atroconidia or hyphae, and not easily disarticulated. Chlamydoconidia are not always in long chain. So here you see one single chlamydoconidia in the hyphae, and here two chlamydoconidia at the tips of hyphae, chain of 4, and very long chain of chlamydoconidia. Okay, there are several dermatophytes that we identify by studying the chlamydoconidia of the dermatophyte, because this dermatophyte, such as Trichobacter verrucosum, we don't see microconidia or macroconidia in Trichobacter verrucosum. So what we normally see is this chain of chlamydoconidia, oftenly referred to as chain of pearls, chain of pearls. And you can also see this terminal vesicle. So under microscope, okay? So this is again a terminal vesicle. So you see terminal vesicle plus chain of chlamydoconidia, which look like chains of pearl. So then you confirm it with characterizing the colony. You describe the colony. So colony of Trichobacter verrucosum, slow growing, small button or disc shapes, white to cream colored, swat light to velvety surface, ray center, and flat periphery with some submerged growth. So reverse pigment may vary from non-pigmented to yellow. Okay, so so this is the description for Trichobacter verrucosum colony. If this fungus grow from skin specimen, nail specimen, and show colony like this, look under microscope and you see chain of pearls, chlamydoconidia, then you can identify the dermatophyte as Trichobacter verrucosum. So actually I have isolated Trichobacter verrucosum many times while working in the lab, and we identify the fungus as T. verrucosum based on characteristic of this chlamydoconidia, because no microconidia, no macroconidia are formed. Okay, this is a post in Elon Musk platform. So, so this person sent this photos, skin lesion, and this is a fungus grew from culturing this lesion, skin scraping, and under microscope, you see a chain of conidia, look like chain of pearl. So he asked maybe he asked his follower, so what is this fungus? And after that, he gave the answer. So Trichophyton verrucosum, pearls, okay? Zoophilic dermatophyte, common cause of ringworm in cattle transmitted by direct contact. So Trichophyton verrucosum. Trichophyton concentricum. So this is another trichophyton which do not produce macro and microconidia in our routine mycological culture used for identifications of mold. So this is the colony of Trichophyton concentricum. And under microscope, you see chlamydoconidia, and the chlamydoconidia has been described as balloon-shaped chlamydoconidia. So if you see balloon-shaped chlamydoconidia, the culture is from skin or nail, and the colony fit to the descriptions of Trichophyton concentricum, then you can report it to the clinician that the fungus is Trichophyton concentricum. If you ask me, I dare to report, even though I don't see the microconidia, no macroconidia, just chlamydoconidia. But based on this characteristic of chlamydoconidia, plus the colonial morphology characteristic, I can happily identify the Trichophyton, the fungus as Trichophyton concentricum. Okay, lastly, we be talking about Aureal Basidium pullulans. This is very unique fungus. This is black yeast fungus. Even though we call it black yeast, but they are mold. They produce hyphae through hyphae, and the hyphae turn into chlamydoconidia, okay, because they are wider than the normal hyphae. They are darker. So this is not atroconidia, but we should call this one chlamydoconidia. So how do we identify Aureal Basidium? First of all, we can see two types of true hyphae. One is hyaline, the other one is dermatiaceous, and the dermatiaceous form chlamydoconidia. And if we look at the hyaline hyphae, it also produces conidia directly from the hyphae. Okay, you can see lots of conidia there directly from the hyphae, and also we can see yeast cells here, blastoconidia. So if you see hyaline hyphae, dermatiaceous hyphae, chlamydoconidia, you see conidia from hyphae, and you see blastoconidia. So this culture is Aureal Basidium pullulans. It's very, very unique because you may think that what you're looking at is mixed culture, not pure culture. No, it is pure culture, but it shows you variety of conidia, and also you can see both hyaline Dermatiaceous hyphae. So this is Aureo Basidium pullulans. Okay, that is my last slide. Thank you for your attention.
Thank you, Dr. Fuat, for the very comprehensive presentation on fungal identification. Actually, I see there's no question in the Q&A chat box. However, there's a attendee who raised their hand, microbiology HWKKS. Do you have any question to ask Dr. Fuad? I have already unmute you. Okay, maybe they don't have any question. Maybe they accidentally raised their hand. Okay, seeing that there are no questions at the moment, we will proceed to conclude this CME session due to the limited time. Just some key points stressed by Dr. Fuad. It's better to verify results from reference lab by at least identifying whether the fungi are adventitious or highline. And also, another key point that was stressed by Dr. Fuad is direct microscopy will aid in identifying and diagnosing the fungal. Okay. So thank you again, Dr. Fuad, for your time and expertise. So we hope that you will find this session beneficial. Please stay tuned for the part 2 of this series, which will be scheduled soon, and we will send out the details via email. Dr. Fuad, do you have any messages before we end this session?
Again, I would like to stress that when we identify most of us, in fact, when I started working to identify mode, when we prepare the slide, look at the microscope, we look for the spores first. So we don't care about the hyphae, we don't care about whether they are broad hyphae or cynocytic. So we jump straight into the spores. Okay, so if we see the spore, doesn't mean you can identify the mode. Sometimes you need to see how the spores are produced, what structure produce it, and how this structure in relation to the hyphae. So it is very important when you prepare a slide to identify mode. Please prepare a slide that show you hyphae plus the spore, and or the best slide is hyphae plus spore plus the cell that produce the spore or conidia. Then only you can identify the mode. Okay? I have received photos from many hospitals asking for my help to identify mode, but they send me photos containing microconidia or macroconidia only, and I can identify the modes, and I ask them, please prepare again new slide so that I can see the hyphae and if there's any special structure that produce the conidia. So it's very important for you to have a slide which show the hyphae. So if no spores are formed, then you can study the hyphae, whether you can see clamp connection, for example, and how if the hyphae is broad. So if the hyphae is broad, it's probably zygomycete, for example. So that is my advice. prepare good slide. Good slide show you hyphae plus the spore. Okay, that is my advice.
Thank you, Doctor. Actually, there's one question by an attendee. It's mentioned that thank you, Dr. Fuat, for the lecture. Would like to ask if Malaysia has ever reported a significant case of basidomycosis. If we preliminarily release report non-sporulating hyphae, pending identification, prior to sending to referral lab, would clinician ignore the fungal isolation?
Can you repeat the question?
They want to ask if there have been any significant case of basidomycosis reported in Malaysia, and if they have released a preliminary report as non-sporulating hyphae, likely basidomycosis, pending identification, prior to sending to referral lab, would clinician ignore the fungal isolation?
Okay, first of all, about the is there any reports of basidomyces in Malaysia? That one, I don't think so. I don't think so. But for cases, yeah, there were cases in Malaysia. So you know, Malaysian is it's quite difficult to write cases. You know, not like people in other country who they like to write cases, okay, and publish it. But Malaysian, I think they should start now to publish clinical cases involving fungus. Your second question is about the you report to the clinician, you give hint to the clinician that the fungus could be basidomyces, but you are waiting for the confirmation from reference lab, and you ask whether the clinician will ignore the fungus. So I think the clinician should, in that case, look at the patient. I mean the clinical conditions of the patient, and whether they should also do a biopsy, for example, look for fungal hyphae in the clinical specimen, and things like that. Okay, because you already give them hint that the fungus could be basidomyces. But again, this finding could be important in immunocompromised patient. If the patient is not immunocompromised, it's probably not important. But again, the lab that isolate the fungus should do direct microscopy. When IMR was doing the diagnostic work for all hospitals in Malaysia, we did direct microscopy for every specimen. If we have enough specimen sent to the lab, and every morning I have to read 30 to 50 QH preparation every day, because this is very important. Because if you see fungal element in the microscopy, then fungus that grow from the specimen could be significant. But if you see characteristic of fungal element in the direct microscopy, and you grow fungus that show similar cellular morphology, cellular characteristic, then definitely we will report the fungus, because definitely it should be significant. So I think that is my comment on that. So I think the clinician shouldn't directly think that the fungus is not significant. But as I said, maybe they have already have the clinical, they have information on the clinical condition.
Correlation. Yeah.
Yeah,
Yeah.
so they said no, this is not fungus, a fungal infection. So they have a right not to ignore the reports of the the fungus. Okay.
Okay. How about non-sporulating hyaline mold from the eyes? Whether it should be investigated further.
I think eyes fungal infection is special, it's unique because many fungus, um, when many fungus, when they infect the eye, and when we culture them on mycological media, they do not form spore. So we will, and if you do direct microscopy, you can see fungal hyphae. So, but when you culture them, it probably show just hyphae. That means we will report the fungus as a non-sporiting mold. But you know, there were cases, aspergillus infecting eye. So then we grew the eye scraping concept, and this aspergillus did not form spore, you know. So it was confirmed aspergillus by DNA sequencing. So lots of question on this non-sporiting mode, whether we should consider it to be significant or not. But I think if you do direct microscopy, that will help you. One way it would help you to determine whether the fungus is significant or not. Okay? Uh, in case if you grow them and they don't produce conidia, but if they produce conidia, then you can identify them. And I think there's a lot of saprophytic fungus can infect the eye, cornea. So I think uh, we should be very careful to determine whether this is significant, isolate or not.
Okay, doctor. Another question by an attendee. I think this is the last question because
Yeah.
we are running out of time. Which part of the mold would you advise to pick from? Which part of the mold would you advise to pick from? And the attendee also for the slide preparation. For slide preparation, which part of the mold would you advise to pick from?
I see. I think the question is which part of colony, I think.
Yes, which part of colony? Yes.
Yeah, I think if you look at one of my slide, the fungus colony grew outward. So the young hyphae will be at the periphery. So spore will be formed on the on the older hyphae. So it would be from the center to maybe midway to the periphery of the colony. Okay. If you pick at the center, the hyphae is probably too old. I think the best point to pick the fungal colony for microscopic preparation is somewhere at the middle, where the hyphae is matured and they probably produce conidia, and the conidia is still in good condition, still in good shape, so you can identify the fungus. But if you prepare slide, if you pick hyphae at the periphery, you probably see only hyphae, no conidia.
Okay. I think that's it for today. Thank you very much again, Dr. Fuat.
You're welcome.
For the attendance, we have already shared the link, and after the end of the session, I think the host would also share a QR code later. So I think a participant can find the link at the Q&A session, or in the chat box. Okay, thank you very much, Dr. Fuat. I
you're
think we
welcome.
will end this session.
Yeah.
Like I mentioned just now, for the part 2 of this series, it will be scheduled soon and we will send out details via email.
Yeah.
Thank you very much, Doctor.
You're welcome.